I wake in the darkness before dawn, my 8 arms coiled beneath me in a crevice between 2 barnacle-encrusted rocks. The water temperature reads just above 13 degrees Celsius through the sensitive cells of my skin, cooler than yesterday, signaling that the current has shifted during the night. My body registers every subtle change: salinity, pressure, the faint vibrations of waves breaking on the reef 60 meters above, the chemical signatures of hundreds of organisms drifting past my shelter. I am an octopus, a member of the order Octopoda, part of the class Cephalopoda, which includes my relatives the squid, cuttlefish, and nautilus. Scientists have described nearly 300 species of octopus worldwide, and I belong to one of the most widespread and well-studied, Octopus vulgaris, the common octopus, though researchers continue to debate whether what we call the common octopus is actually several distinct species with overlapping ranges and subtle genetic differences. My body is unlike that of almost any other animal. I have no bones, no rigid skeleton of any kind. The only hard structure in my entire body is my beak, a sharp parrot-like structure made of chitin, tucked in the center where my 8 arms meet. This means I can squeeze through any opening larger than that beak, compressing and elongating my soft tissues to fit through gaps that seem impossibly small. My mantle, the muscular sac that contains most of my internal organs, measures about 20 centimeters from front to back at this stage of my life, though members of my species range from 10 to 45 centimeters in mantle length depending on age, sex, and geographic population. My arms extend much farther, each one roughly 4 to 5 times the length of my mantle, giving me a total span of nearly 1 meter from arm tip to arm tip when I stretch fully. I weigh approximately 3 kilograms now, though the largest individuals of my species can reach 10 kilograms or more in regions with abundant food and few predators. Each of my 8 arms is lined with suckers, 2 rows running the entire length, more than 200 suckers per arm, 1,600 or more across all 8 limbs. Each sucker operates independently, controlled by its own neural cluster, its own ring of muscle that can contract to create powerful suction or relax to release. The suckers are not merely suction cups for holding. They are sense organs, perhaps the most sophisticated tactile and chemical sensors in the animal kingdom. Each sucker contains thousands of chemoreceptors that allow me to taste everything I touch. When I probe a crevice in the rock, my suckers analyze the chemical signature of the surface, detecting the faint residue of a crab that passed hours ago, the mucus trail of a snail, the metabolic byproducts of bacteria growing on algae. I taste the world in 3 dimensions, my arms extending my awareness into spaces my eyes cannot see. My skin is an organ of transformation. At this moment, in the shadowed security of my den, my color is a muted brownish red stippled with white, matching the algae and coralline growth on the surrounding rocks. This is not a conscious choice in the way a human might choose clothing. The process occurs through a system of specialized cells called chromatophores, layered in my skin like pixels in a screen. Each chromatophore is a tiny elastic sac filled with pigment, red or yellow or brown, surrounded by a ring of muscle cells. When the muscles contract, the sac expands, displaying the color. When they relax, the sac contracts to a pinpoint, hiding the color. Beneath the chromatophores lie iridophores and leucophores, cells containing structures that reflect light, producing iridescent blues and greens and whites. I can activate different combinations of these cells across different patches of my skin in less than a second, transforming my appearance completely. I do this not by thinking about what color I want to be, but through a complex interaction between my vision, my brain, and neural pathways that bypass conscious control. I see my surroundings, and my skin responds, sometimes before my brain has fully processed what my eyes have detected. The transformation extends beyond color. My skin can change texture as well. Beneath my chromatophores, I have muscular structures called papillae that I can raise and lower, creating bumps, spikes, ridges, and smooth patches to match rocks, coral, sand, or seaweed. Right now, in my rocky den, my skin displays small, irregular bumps that mirror the texture of the stone around me. When I venture onto sand later, I will smooth these away and adopt a different pattern entirely. As the dim light of early morning begins to filter down through the water, I shift position slightly, and a small fish, a juvenile wrasse, darts past the entrance to my shelter. My eyes track its movement with precision. My eyes are large, sophisticated organs, superficially similar to vertebrate eyes but evolved entirely independently, a remarkable case of convergent evolution. I have a lens, a retina, and an adjustable pupil, just as a human does, but the underlying structure and development are completely different. My retina has no blind spot. The arrangement of photoreceptor cells is more uniform, and I can detect polarized light, something most mammals cannot do. Polarization vision helps me detect transparent prey like jellyfish and see through the reflective camouflage of silvery fish. My pupil is a distinctive horizontal rectangle that never fully closes, allowing me to monitor my surroundings even when I rest. I do not have color vision in the traditional sense. I possess only one type of photoreceptor pigment, which would normally mean I see the world in shades of one color. Yet evidence suggests I may detect color through other means, possibly by using chromatic aberration, the way different wavelengths of light focus at slightly different distances, or through skin-based light sensors that exist independently of my eyes. Researchers are still studying this. I emerge from my den as the morning brightens, moving with a grace that seems alien to creatures accustomed to limbs and joints. I do not walk so much as flow. My arms extend and contract in coordinated waves, their suckers gripping rock and pulling me forward while my mantle pulses gently to adjust my buoyancy. If I need to move quickly, I have another method entirely: jet propulsion. I draw water into my mantle cavity through openings on either side of my head, then close those openings and contract my entire mantle forcefully, expelling the water through a muscular funnel called a siphon. The siphon is flexible, and I can aim it to push myself forward, backward, or to the side. This method is fast but energetically expensive, burning through the limited oxygen my blood can carry. I use it for short bursts, escaping from threats or lunging toward prey, but for routine movement I rely on my arms. The oxygen-carrying capacity of my blood is inherently limited because I use hemocyanin rather than hemoglobin as my respiratory pigment. Hemocyanin, a copper-based molecule, turns my blood pale blue when oxygenated, unlike the iron-based hemoglobin that makes vertebrate blood red. Hemocyanin is less efficient than hemoglobin at binding oxygen, especially in warm or acidic conditions, which means I am sensitive to water temperature and cannot sustain high activity levels for long. This is one reason most octopuses, including my species, live in relatively cool waters or restrict activity during the warmest parts of the day in tropical regions. My three hearts work constantly to push this sluggish blood through my body. Two branchial hearts pump blood through my gills, located inside the mantle cavity, and one systemic heart pumps oxygenated blood to the rest of my body. When I use jet propulsion, the systemic heart actually stops beating, which is why I cannot maintain that mode of travel for more than a few seconds before I must rest. I move across the reef, my skin shifting continuously. When I cross a patch of white sand, I pale, flattening my papillae and spreading out my arms to minimize my shadow. When I encounter a section of reef covered in brown and purple algae, my skin erupts in matching colors and bumps. A passing grouper, a predator large enough to swallow me whole, glides overhead. I freeze instantly, not just stopping movement but locking my muscles into absolute stillness, because motion attracts attention far more than color mismatch. The grouper continues without pausing, its attention on a school of smaller fish in the midwater. Once it passes, I resume my hunting. I am searching for crabs. Crabs are a favored prey for many octopuses, including my species, though our diet varies widely depending on what is available. I consume crustaceans of many kinds, including shrimp, lobsters, and hermit crabs, as well as mollusks like clams, mussels, and occasionally other octopuses or small squid. I eat fish when I can catch them, typically slow-moving or sleeping individuals, and I take worms, snails, and other invertebrates opportunistically. My hunting strategy depends on what I am hunting. For mobile prey like crabs, I rely on stealth and speed. For immobile prey like clams, I rely on patience, strength, and chemistry. A flicker of movement catches my eye. A small crab, perhaps 4 centimeters across its carapace, scuttles across a rock 5 meters ahead. I lower my profile, tucking my arms close and allowing my skin to match the substrate perfectly. Then I begin to stalk. I move in slow pulses, freezing between each advance, keeping my body oriented so the crab remains in my field of view. My eyes are positioned on the sides of my head, giving me nearly 360 degrees of vision, though I focus best on objects directly in front of me. As I close the distance, I gather my arms beneath me like springs. When I am within one body length, I explode forward, arms spreading like a web, suckers outstretched. The crab tries to dart away, but 2 of my arms intercept it, suckers latching onto its shell and legs. I draw it toward my mouth, which is located on the underside of my body at the center point where all 8 arms converge. My beak bites through the joint in the crab's carapace with a cracking sound that travels through the water as a sharp click. I inject saliva into the wound, saliva that contains a complex mix of enzymes and toxins. The toxins, some derived from bacteria that live symbiotically in my salivary glands, paralyze the crab's nervous system within seconds. The enzymes begin breaking down proteins, pre-digesting the flesh before it enters my digestive system. I spend several minutes manipulating the crab, my beak tearing away sections of shell, my arms sorting edible tissue from indigestible fragments. I swallow the soft parts in small pieces. My esophagus passes directly through the middle of my brain, so large chunks could cause neural damage. This anatomical quirk is one of many oddities that result from the molluscan body plan being adapted to an active predatory lifestyle, so different from that of my slow-moving ancestral relatives like snails and clams. After feeding, I move toward a different section of reef where the rocks form deeper crevices. I am looking for a new den, a safer place than the exposed crack where I spent last night. I am particular about dens. A good den has a narrow entrance that I can block, a space inside large enough to turn around, good water flow to bring oxygen and remove waste, and multiple escape routes if possible. I inspect several candidates, reaching my arms deep inside to taste the interior, checking for the presence of other occupants, moray eels or other octopuses or territorial fish. I find one that meets my requirements. A cavity beneath a boulder, the entrance the size of my fist, the interior space about twice the volume of my body. I squeeze inside, my body compressing and elongating, my mantle folding, my arms threading through the gap in a sequence that looks impossible. Once inside, I arrange myself comfortably, my arms coiled, my eyes positioned to watch the entrance. The space outside my den entrance is littered with shell fragments, crab legs, and broken bits of sea urchin, the remains of meals from previous occupants or perhaps my own meals that I will add to over time. Octopuses are known for creating middens, piles of debris outside their dens, and researchers sometimes use these middens to study diet without directly observing feeding behavior. I gather several stones from inside the den and use my arms to position them near the entrance, not fully blocking it but creating a barrier I can adjust. Some octopuses are known to manipulate objects extensively, carrying coconut shells or clam shells as portable shelters, stacking rocks into walls, even throwing stones or shells at intruders. This behavior is documented but not universal. It varies by individual and population, and scientists continue to debate the extent to which it represents tool use in the strict definition. Settled into my den, I enter a state of rest, though I do not sleep in the way mammals do. My activity level drops, my color fades to a dull mottled pattern, and my arms relax. But my eyes remain open, my pupils tracking movement outside, and my suckers maintain a light grip on the rocks around me, monitoring vibrations. Octopuses may experience something like sleep, including periods where the brain shows different activity patterns and the body displays color changes that might correspond to dreaming, but the evidence is still preliminary. What is clear is that I have periods of rest and periods of activity, typically aligned with the light cycle, though my species is most active during dawn and dusk, a pattern called crepuscular. As I rest, I remain aware of the chemical environment. My skin, my suckers, and specialized sensory cells throughout my body constantly sample the water, detecting dissolved substances at concentrations far below what most organisms can sense. I can smell, though not in the way a land animal smells air. I detect amino acids, the building blocks of proteins, which leak from the bodies of all animals. I detect nucleotides, the molecules that leak from damaged cells, signaling injury or death. I detect pheromones, chemical signals released intentionally by other animals for communication. The ocean is a soup of information, and I taste all of it. Hours pass, and the light begins to shift as the sun arcs higher. The water temperature rises slightly, now nearing 15 degrees Celsius. I can feel the change through my skin, which is not merely a surface covering but an integrated sensory system that monitors temperature, pressure, water flow, and chemical composition continuously. My body temperature matches the surrounding water almost exactly. I am ectothermic, lacking the internal heat generation that mammals and birds use to maintain constant body temperature. This means my metabolic rate rises and falls with environmental temperature, speeding up in warmer water and slowing in colder. In tropical populations of my species, individuals may be significantly more active, but also have shorter lifespans, burning through their physiological resources more quickly. In colder waters like those where I live now, growth is slower but total lifespan may be longer, though predation and other mortality factors complicate this pattern. A shadow passes across the entrance to my den, and I focus both eyes forward. A small shark, perhaps a dogfish, cruises past, its electroreceptors scanning for prey buried in the sand. Sharks are among my predators, though not the most significant. Larger fish pose the greatest threat. Groupers, snappers, eels, and rays all consume octopuses regularly. Marine mammals, including seals and dolphins, hunt us with intelligence and persistence. Sea otters in some regions specialize in extracting octopuses from dens, using rocks to break shells and reach hidden prey. Sea birds, particularly when we venture into shallow water, can strike from above. Even other cephalopods, large squid and cuttlefish, may attack smaller octopuses. And of course, other octopuses are cannibalistic. A larger individual will consume a smaller one without hesitation if the opportunity arises. My defenses against these threats are varied and sophisticated. Camouflage is the first line, allowing me to avoid detection entirely. When camouflage fails, I have other options. I can perform dramatic displays, suddenly expanding my mantle, spreading my arms wide, and flashing high-contrast patterns of dark and light, sometimes forming eye spots on my body that make me appear larger or more dangerous. I can release ink, a dark cloud of melanin particles mixed with mucus and other chemicals, which not only obscures vision, but also may dull the predator's sense of smell, and even contain compounds that irritate sensitive tissues. The ink cloud can hang together as a decoy roughly the size and shape of my body, allowing me to jet away while the predator attacks the phantom. I can also release the ink as a diffuse smokescreen and use my siphon to create turbulence that spreads the cloud, giving me time to hide and change color before the water clears. If a predator grabs one of my arms, I can autotomize it, severing the arm at a predetermined breaking point through muscular contraction and allowing the predator to take the limb while I escape. The severed arm continues to move for minutes or even hours after separation, its autonomous nervous system still functional, its suckers still reacting to stimuli. This continued movement can distract the predator while I retreat. Over weeks or months, I can regenerate the lost arm, growing it back from the wound site, though the regenerated limb may be slightly shorter or have fewer suckers than the original. Autotomy is costly, reducing my hunting efficiency and making me more vulnerable to subsequent attacks, so I use it only as a last resort. My nervous system is unlike that of any vertebrate. My brain, located between my eyes, contains approximately 500 million neurons, a number comparable to that of a dog, though organized entirely differently. But more than half of my neurons are not in my brain at all. They are distributed through my arms, with each arm containing its own nerve cord and neural ganglia that process information and control movement semi-independently. When I send a command from my brain to move an arm toward a target, I do not micromanage each sucker and each muscle contraction. Instead, my brain sends a general instruction, and the arm's own neural network executes the details. This distributed intelligence allows me to perform multiple complex tasks simultaneously, with different arms solving different problems at the same time. One arm exploring a crevice, while another manipulates an object, while a third maintains my grip on the substrate. This creates a unique relationship between my brain and my body. I do not have complete awareness of what each arm is doing at every moment. An arm may discover food while exploring and begin manipulating it before my brain has fully registered the find. Researchers have documented octopuses whose arms reach for food, while the animal's attention is focused elsewhere, suggesting that the arms possess something approaching independent decision-making capacity. Whether this constitutes a form of distributed consciousness or simply reflects sophisticated reflex arcs remains a topic of scientific debate. What is clear is that my subjective experience of having a body is fundamentally different from that of animals with centralized nervous systems. The water current shifts again, bringing new chemical signals. I detect the scent of another octopus, a faint trace of amino acids and hormones that indicates a female passed through this area recently, perhaps within the last hour. I am male, identifiable by the modification of my third right arm, which is specialized for reproduction. The tip of this arm, called the hectocotylus, has a modified structure with a groove running along its length and fewer suckers than my other arms. During mating, I will use this arm to transfer packets of sperm, called spermatophores, to the female. But I am not yet ready to breed. I am perhaps 18 months old, approaching maturity but not fully mature, and the breeding season for my population typically peaks in late spring and early summer, several months from now. Octopus mating systems vary by species and by population. In my species, Males and females are generally solitary, coming together only briefly for mating. Males compete for access to females, and larger males typically dominate smaller ones, though smaller males sometimes use alternative strategies, adopting female-like coloration and behavior to sneak past dominant males and achieve matings. Courtship involves visual displays, the male showing pulsing color patterns and approaching cautiously, because the female may attack and consume him if she is not receptive. If she accepts, the male extends his hectocotylus into her mantle cavity, depositing spermatophores near her oviduct. The process can take an hour or more, and the male may mate with multiple females over his lifetime, though each mating represents a significant investment of energy and risk. Females store the sperm internally and may delay fertilization for weeks or even months, waiting for optimal conditions to lay eggs. When she is ready, she seeks out a suitable den, somewhere protected and secure where she can remain for the entire brooding period. She lays her eggs in long strings, each egg about the size of a grain of rice, thousands or tens of thousands of eggs depending on her size and condition. She uses a secretion to glue the eggs to the roof of her den, hanging them in clusters that resemble bunches of grapes. Once the eggs are laid, her life changes completely. She remains in the den, guarding the eggs, grooming them with her arms and flushing them with clean water from her siphon to prevent fungal growth and ensure adequate oxygen. She stops eating. This is not a choice. Her body undergoes hormonal changes that suppress her appetite and redirect all her energy toward egg care. For 1 to 2 months, depending on water temperature, she tends the eggs constantly, growing weaker, her muscles wasting, her organs beginning to fail. When the eggs hatch, releasing thousands of tiny paralarvae into the water, she dies, her body exhausted beyond recovery. This pattern, called semoparity, in which an organism reproduces once and then dies, is characteristic of all octopuses. We live fast, grow quickly, reproduce once and die young. Males typically die shortly after mating as well, though the decline is less abrupt. After successful reproduction, males often become less cautious, more active, and more vulnerable to predation. Their bodies also begin to deteriorate, a process driven by hormones released from the optic glands, small endocrine structures located near the eyes. Research has shown that removing the optic glands can extend an octopus's lifespan significantly, allowing individuals to live beyond their natural reproductive terminus, though this intervention prevents normal reproduction. The optic glands appear to function as a programmed death mechanism, ensuring that adults die after reproduction, a pattern whose evolutionary logic likely relates to the energetic demands of our lifestyle and the ecological niches we occupy. My species typically lives 1 to 2 years from hatching to death, though some individuals in favorable conditions may reach 3 years. The paralarvae that hatch from eggs are tiny, measuring only a few millimeters, transparent and delicate, drifting in the plankton. This planktonic stage lasts for weeks or months, depending on water temperature and food availability. The paralarvae feed on microscopic zooplankton, growing gradually, molting their skin periodically as they increase in size. Most die during this vulnerable stage, consumed by planktivorous fish, jellyfish, and other predators. The few that survive eventually descend to the sea floor and transition to the benthic lifestyle, settling into crevices and beginning to hunt larger prey. At this stage, they are still very small, perhaps a centimeter in mantle length, and they face a new suite of predators. Growth is rapid if food is abundant. A young octopus can double its weight in a matter of weeks. By 1 year of age, most individuals have reached a significant portion of adult size, though growth continues until reproduction. I do not know my parents. There is no parental care after hatching, no teaching, no family structure. Everything I know is either instinctive, encoded in my genes, or learned through individual trial and error. Yet I am capable of remarkably sophisticated learning. Octopuses can solve puzzles, learn to open containers with screw-top lids or latches, remember solutions over weeks or months, and distinguish between individual humans based on their behavior. We can learn by observation, watching another octopus solve a problem, and then solving it ourselves more quickly. We can adapt our behavior based on context, using different hunting strategies for different prey types, adjusting our camouflage to match new environments we have never encountered before. This learning capacity exists despite my short lifespan and the absence of any social learning structure, which makes it unusual compared to other animals with similar cognitive abilities. The intelligence of octopuses has fascinated and puzzled scientists since systematic study began. We possess cognitive abilities that rival or exceed those of many vertebrates, yet our evolutionary lineage diverged from the vertebrate line more than 500 million years ago, long before the evolution of fish. Our intelligence evolved independently, shaped by different pressures, built on a different neural architecture. Where mammalian intelligence is centralized in a large brain with specialized regions for different functions, my intelligence is distributed, embedded in a body plan that treats each arm as semi-autonomous. Where birds and mammals have long lives and extended parental care that allows for social learning, I must learn everything alone in a lifespan shorter than that of many insects. The evolutionary pressures that produce this intelligence likely include the need to handle diverse prey types that require different manipulation strategies, the need to navigate complex three-dimensional reef environments, and the intense predation pressure that favors rapid learning and behavioral flexibility. The sun reaches its zenith, and the reef becomes busier. Schools of small fish drift past, feeding on plankton in the water column. A sea turtle surfaces nearby, its movement slow and graceful. I remain in my den, my color settled into a pattern that matches the interior shadows, dark brownish red with scattered white spots. I am not hungry yet. The crab I consumed earlier will sustain me for hours. My metabolism is efficient but not particularly fast compared to active fish. I do not maintain high activity levels constantly, instead alternating between periods of active hunting and long periods of rest or slow movement, a strategy that conserves energy in an environment where food availability is unpredictable. As I rest, I perform maintenance behaviors. I run my arms over my mantle, cleaning the skin, removing debris and parasites. Octopuses can host various parasites, including copepods that attach to the skin and gills, protozoans that infect internal organs, and worms that encyst in tissues. I groom regularly to minimize external parasites, using my suckers to detect and remove anything that does not belong. I also flush water through my mantle cavity, ensuring that my gills remain clean and free of sediment. My gills are delicate structures, feathery and highly vascularized, where gas exchange occurs. If they become clogged with debris, my oxygen intake drops, and I become sluggish and vulnerable. A Nassau grouper, a large predator perhaps 80 centimeters long, takes up a position on a rock outcrop 20 meters from my den. This is concerning. Groupers are intelligent, patient hunters with excellent vision and the capacity to learn the locations of octopus dens. They often station themselves near a den and wait for the occupant to emerge, or they probe the entrance with their snouts, trying to flush the octopus out. I do not move, my color locked in camouflage, my arms tucked tightly against my body. The grouper scans the reef, its eyes moving independently. Minutes pass. Eventually, the grouper swims off to investigate another section of reef, and I relax slightly. But I make a mental note: this den is known to predators. I may need to relocate again soon. The concept of a mental note in a creature like me requires some explanation. I do have memory, both short-term and long-term. I can remember the locations of dens, the routes I have traveled, the types of prey I have encountered in different areas, and the appearance of threats. But my memory is not organized in the narrative, episodic way that humans experience memory. I do not reminisce or plan far into the future. My memory is functional, linked directly to behavior. This place had food, this route is dangerous, this shape means predator. The question of whether I have subjective experiences, whether there is something it is like to be me, is one that science cannot yet answer definitively. I behave in ways that suggest I experience pain, fear, curiosity, and perhaps something like frustration or satisfaction. But whether these behaviors reflect internal experiences comparable to human emotions or are simply sophisticated behavioral algorithms remains unknown. What is clear is that I am capable of behavioral flexibility that goes beyond simple stimulus-response reflexes. If I try to open a container to reach food and fail, I try different approaches, adjusting my strategy based on what does and does not work. If a particular den proves unsafe, I abandon it and search for alternatives. If a predator begins hunting me, I do not always use the same escape behavior. I vary my response, sometimes fleeing, sometimes hiding, sometimes displaying, depending on the context. This flexibility is a form of intelligence, whatever its underlying mechanism. As afternoon progresses into evening, the light begins to dim, and the reef transitions to its nocturnal state. Fish that were active during the day retreat to crevices to sleep. Nocturnal species begin to emerge. Moray eels slide from their caves, their serpentine bodies undulating through the rocks. Lobsters emerge from crevices, their antennae twitching as they search for food. A small octopus, possibly a juvenile of my own species, appears on a nearby ledge, its skin showing the smooth texture and pale color typical of younger individuals. The appearance of the juvenile triggers a response in me, not protective or social, but predatory. I am large enough to consume that juvenile, and octopuses are opportunistic cannibals. But the juvenile is wary, remaining at a distance, its eyes watching me. It moves away along the reef, and I do not pursue. I am not hungry enough to expend the energy on a chase, and the juvenile's alertness suggests it would be difficult to catch. I leave my den as twilight deepens, my body shifting to a darker pattern appropriate for low-light conditions. I move across the reef with more confidence than during the day, emboldened by the reduced visibility that protects me from visual predators. My nocturnal hunting begins. I am searching now not for crabs but for sleeping fish, parrotfish that wedge themselves into crevices and secrete mucus cocoons around their bodies, wrasses that tuck themselves into the sand, small groupers that rest in the open, their reflexes slowed by sleep. Sleeping fish are easier prey than active ones, less able to detect my approach, slower to react. I find a parrotfish in a narrow crack, its body covered in the transparent mucus cocoon that masks its scent from nocturnal predators. The cocoon is effective against some predators but not against me. I can still detect the fish through chemical traces, and the cocoon provides no physical protection. I reach into the crack with 2 arms, my suckers gripping the rock for leverage while the tips of my arms probe the cocoon. The parrotfish wakes and struggles, but it is trapped in the confined space. My arms surround it, pulling it from the crack. My beak finds the base of its skull and bites through the vertebrae, severing the spinal cord. The fish goes limp. I spend the next 20 minutes consuming it, tearing away scales and skin, pulling off flesh in small pieces that I can swallow safely. After feeding, I continue moving across the reef, not hunting now but exploring. Octopuses are curious animals. investigating novel objects and environments even when there is no immediate benefit. This curiosity may be an extension of foraging behavior, a tendency to explore that increases the probability of finding food or dens, or it may serve other functions related to spatial learning and cognitive mapping. I encounter a broken piece of pottery, probably debris from a human boat, and I manipulate it with several arms, turning it over, tasting it with my suckers, determining that it is inedible but potentially useful. I carry it back toward my den, considering whether it might serve as a door or wall. But the shape is awkward, and I eventually abandon it in favor of natural rocks. The moon rises, nearly full, its light penetrating the water and creating shifting patterns of brightness and shadow on the reef. My eyes are highly sensitive to light, capable of functioning in conditions that would render most humans effectively blind, and the moonlight is more than sufficient for me to see clearly. I notice other octopuses moving on the reef, distant shapes that I recognize by their movement patterns, the distinctive way arms flow over rocks, the sudden color changes. My species is generally solitary, maintaining individual home ranges that may overlap, but within which direct interactions are rare except during mating season. When two octopuses encounter each other outside of reproductive context, the interaction is usually brief and agonistic, a contest to determine which individual will retreat. Size is the primary determinant of dominance. Larger individuals typically prevail, and smaller ones withdraw. I encounter a conspecific, another octopus slightly smaller than myself, emerging from a den opening. We see each other simultaneously, and both of us immediately display. My skin flushes with dark patterns, and I spread my arms wide, raising my body high on my arm tips to maximize my apparent size. The other octopus mirrors the display. We remain frozen in this posture for several seconds, each assessing the other. I am marginally larger, and perhaps my display is more intense because the other octopus suddenly changes tactics, its skin paling and smoothing, its body lowering into a submissive posture. It retreats into its den. I do not pursue. The contest is resolved without physical contact, a common outcome when the size difference is perceptible. I move on, crossing a sandy patch toward another section of reef. In the open sand, I adopt a completely different strategy. I bury myself, using my arms to excavate sand and pull it over my mantle, leaving only my eyes exposed, my skin textured and colored to match the sand grains perfectly. I become a bump in the sand, invisible to passing predators. A small ray glides past, its electroreceptors scanning the sand for buried prey, but it does not detect me. Octopus muscle and nerve tissue do produce electrical signals, but the signals are weak, especially when I am motionless, and my body lacks the strong electrical signature of fish or crustaceans. The ray passes over me without pausing. From my buried position, I watch the nighttime reef. A Caribbean reef squid hovers in the water column above me, its fins rippling, its tentacles extended. Squid are relatives, fellow cephalopods, but our lifestyles differ significantly. Squid are pelagic, living in open water, hunting with speed and vision, often operating in coordinated groups. I am benthic, tied to the sea floor, a solitary ambush predator. We share a common ancestor, but our lineages diverged hundreds of millions of years ago, and our bodies reflect different evolutionary solutions to different problems. The squid's body is streamlined for sustained swimming, its arms and tentacles arranged around a forward-facing mouth, its mantle forming a hydrodynamic torpedo shape. My body is optimized for flexibility and camouflage, my arms arranged radially around a central mouth, my mantle a flexible sac that I can distort into almost any shape. A boat passes on the surface far above, its engine a low rumble that I feel through the water as pressure waves. Humans are an increasing presence in the ocean. Commercial fishing boats drag nets and deploy traps that kill octopuses as bycatch, even when we are not the target species. Recreational divers and snorkelers seek us out, fascinated by our color changes and intelligence. Scientists capture us for research, studying our neurobiology, our cognition, our behavior. Humans also consume us in large quantities. Octopus is a valuable seafood in many cultures, and fishing pressure on some populations is intense. My species is widely distributed and not currently considered endangered overall, but local populations in heavily fished areas have declined significantly, and there is growing concern about sustainability. The relationship between humans and octopuses is complex. In some cultures, we are regarded with suspicion or fear, seen as alien and potentially dangerous. In others, we are celebrated for our intelligence and adaptability, featured in art and folklore. Ancient Greek and Roman sources described octopuses as clever animals, capable of solving problems and escaping from containers. Pacific Island cultures have traditional stories about octopuses as trickster figures or shapeshifters. Modern media portrays us sometimes as monsters, sometimes as sympathetic characters, often emphasizing our otherworldliness. The reality is more nuanced. We are intelligent, but not in the way mammals are intelligent. We are adaptable, but also constrained by our physiology and short lifespans. We are neither monsters nor friends. We are animals, shaped by evolution to fill particular ecological roles. Human impact on octopus populations extends beyond direct fishing. Climate change is altering ocean temperatures, and because we are ectothermic, even small changes in temperature can significantly affect our metabolism, growth rates, reproductive timing, and survival. Ocean acidification, caused by increased carbon dioxide absorption, may affect our eggs and paralarvae, which are more sensitive to pH changes than adults. Coastal development destroys reef and rocky habitats that we depend on for dens. Pollution introduces toxins that accumulate in our tissues. As predators relatively high in the food web, we concentrate pollutants from all the prey we consume. Plastic debris litters the ocean, and octopuses sometimes use discarded bottles or containers as dens, a behavior that seems adaptive but may expose us to harmful chemicals leaching from the plastic. Despite these threats, octopuses as a group are relatively resilient compared to many marine animals. Our short lifespans and high reproductive output mean populations can recover quickly if conditions improve. We are generalist predators, capable of switching prey types when preferred species are unavailable. We can tolerate a relatively wide range of environmental conditions, and some species are expanding their ranges as ocean temperatures rise. But resilience has limits, and the cumulative effects of multiple stressors, fishing pressure combined with habitat loss combined with climate change, may eventually overwhelm our adaptive capacity. I emerge from the sand as the night deepens, shaking the grains from my skin, and continue my exploration. I am moving toward the edge of the reef, where it drops off into deeper water. The slope is steep here, descending from 30 meters to 60 meters within a short distance. I pause at the edge, my arms gripping the rocks, my eyes scanning the darker water below. Deep water is unfamiliar territory for me. I typically remain in shallower areas where light penetrates and reef structure provides abundant hiding places. But curiosity and the possibility of unexploited food resources draw me downward. As I descend, the water cools noticeably, dropping from 15 degrees to 12 degrees Celsius. My metabolism slows in response, my movements becoming slightly less fluid, my reaction time lengthening. The pressure increases as well, though this is not a significant stress for me. My body is mostly water and soft tissue, containing no gas-filled spaces except for a small amount of air in my digestive tract, so I do not experience pressure-related problems the way diving mammals do. I can move from the surface to 100 meters depth without difficulty, and some octopus species live at depths exceeding 3,000 meters, in the abyssal zone where pressure reaches hundreds of atmospheres. The deep reef is different from the shallows. The coral gives way to rock covered in sponges and soft corals. The light is dimmer, even with the moon above. At 60 meters, only the blue-green wavelengths of light penetrate in significant quantities, and my surroundings take on a monochromatic appearance. My color vision, already limited, becomes almost irrelevant here. I navigate more by texture and shape than by color. Bioluminescence becomes more common. I see the faint glow of bacteria on decaying organic matter, the brief flashes of dinoflagellates disturbed by my movement, the luminous lures of anglerfishes waiting motionless for prey. I encounter a lobster, a large spiny lobster, its antennae longer than my entire body. Lobsters are formidable prey, armed with a tough exoskeleton and powerful tail that can propel them backward in rapid escape maneuvers. But they are also nutritious, worth the effort I approach cautiously, trying to position myself to grab the lobster's body behind the antennae where it cannot easily retaliate. The lobster detects me, its antennae whipping around, and it begins to retreat, walking backward on its legs. I lunge, arms extended, but the lobster flips its tail and rockets backward, colliding with a rock. I pursue, but the lobster squeezes into a crevice too narrow for me to enter. I spend several minutes probing the opening with my arm tips, trying to reach the lobster, but it has wedged itself securely. I abandon the attempt. Hunting is not always successful. Prey animals have their own defenses, their own strategies, and a significant percentage of my attacks fail. This is why I must hunt frequently, taking multiple prey items per day to meet my energy needs. My energy budget is constantly precarious, balanced between intake from food and expenditure on movement, camouflage, cognition, and basic metabolism. If I go too long without food, my condition deteriorates rapidly. If I expend too much energy in unsuccessful hunts, I weaken. The margin for error is small. I find a different prey item, a sleeping fish tucked into a crack, and successfully capture and consume it. Then I begin the ascent back to shallower water, preferring the familiar reef over the alien deep. As I climb, I notice the sky beginning to lighten in the east. Dawn is approaching. I have been active all night, and my body is signaling the need for rest. I return to the den I occupied earlier, checking first to ensure the grouper is not waiting nearby. The area appears clear. I squeeze back inside, rearrange my barrier of stones, and settle in. My skin fades to a muted rest pattern, irregular blotches of brown and tan. My arms coil loosely, my mantle relaxes, and my eyes half close, though the rectangular pupils remain partly open, monitoring the entrance. My 3 hearts slow to a resting rhythm. My breathing rate, the contraction of my mantle that draws water over my gills, decreases. I enter a period of quiescence, not full sleep but reduced activity, my brain processing the information gathered during the night, consolidating memories, maintaining neural networks. As I rest, color patterns ripple across my skin, waves of light and dark moving from my mantle down my arms. This phenomenon has been observed in octopuses and may represent something analogous to dreaming, neural activity that plays out through the skin's chromatophore system. Whether I am experiencing anything subjective during these episodes is unknown. The patterns may be random noise, or they may reflect the brain replaying and organizing sensory experiences. There is no way currently to determine which. The morning brightens. Small fish venture out from their sleeping places and resume feeding. The reef shifts from its nocturnal cast to its diurnal cast of species. I remain in my den through the morning and into early afternoon, resting, conserving energy. Octopuses are not strictly nocturnal or diurnal, but rather crepuscular and opportunistic, most active during twilight periods but willing to hunt at any time if a good opportunity presents itself. The pattern varies by individual, by local predator and prey dynamics, and by environmental conditions such as water clarity and moon phase. In late afternoon, I begin to feel restless. The chemical signature of food has been drifting past my den, traces of injured fish, the scent of crabs molting. Molting crabs are particularly vulnerable, their new exoskeleton still soft and unprotected, and they emit distinctive chemical signals that predators like me can detect from considerable distances. I exit my den and begin to hunt, following the scent gradient, my arms testing the water direction to determine the source of the signal. I find the molting crab hidden under a ledge, its old exoskeleton discarded nearby, its new shell still pale and pliable. It attempts to retreat deeper into the crevice, but its movements are slow and uncoordinated, its muscles not yet accustomed to the new dimensions of its body. I reach in and extract it easily. My beak penetrates the soft shell without resistance. The meal is rich, providing not only protein and fat but also trace minerals and vitamins essential for my health. After feeding, I engage in an activity that might seem purposeless. I manipulate a small rock, passing it from arm to arm, rolling it across my suckers, examining it from every angle. This behavior, object play, has been documented in octopuses and other cephalopods and is difficult to explain in purely functional terms. The rock is not food, not a tool I am currently using, not a threat. Yet I spend several minutes interacting with it before eventually discarding it. Possible explanations include that play serves a developmental function, helping young animals practice motor skills and sensory integration, or that it reflects excess cognitive capacity, a brain evolved for complex problem-solving occupying itself when immediate survival tasks are not pressing. Or it may simply feel rewarding, activating neural reward pathways that evolve to encourage exploration and manipulation, behaviors that increase long-term survival even if they have no immediate payoff. I encounter another octopus, this time a female, recognizable by the absence of the hectocotylized third right arm that marks males. She is slightly smaller than I am but appears mature. We approach each other cautiously. I change my color pattern, displaying waves of dark and light that sweep across my body. She responds with her own color changes. This is communication, though not language in the human sense. The patterns convey information about size, health, motivation, reproductive state. I am signaling that I am male, mature, or nearly so, interested but not aggressive. She signals caution, her pattern flickering between receptive and defensive. I extend one arm slowly, not the hectocotylus but an ordinary arm, touching her gently. She does not retreat but does not fully accept either. Her arm touches mine, and we remain in contact for perhaps 30 seconds, suckers tasting each other's skin, sampling chemical signatures that convey genetic compatibility, health status, parasite load, recent diet. This tactile assessment is critical. Octopuses rely heavily on chemotactile information to evaluate potential mates. But something in the exchange is not satisfactory to one of us, or perhaps the season is not quite right, because she withdraws, her color shifting to a pattern that signals disinterest or rejection. I do not pursue. Forced mating is not a successful strategy for octopuses. The female is fully capable of defending herself, and an unwilling female may attack and injure or kill a male who persists. I move away, resuming my solitary routine. The days and weeks that follow establish a pattern. I hunt during twilight and darkness, consuming crabs, fish, mollusks, shrimp, and occasional other prey. I rest during the brightest parts of the day. I move between several dens, rotating among them to minimize predator learning. I avoid threats when possible, defend myself when necessary. I continue to grow, adding mass steadily, my arms lengthening, my mantle expanding. I am approaching full maturity, the point at which reproduction will become the dominant drive, overriding caution and self-preservation. I encounter humans more frequently as time passes. Divers appear on the reef, their bubbles rising in streams toward the surface, their movements clumsy compared to the fish and marine animals around them. They carry cameras, lights, and other equipment. When they spot me, they often approach, extending hands or cameras toward me. My response depends on context and individual personality, a factor that varies among octopuses just as it does among mammals. Some octopuses are bold, approaching humans, investigating their equipment, sometimes reaching out to touch divers. Others are shy, retreating immediately and hiding. I am somewhere in the middle, cautious but curious. When a diver approaches slowly and non-threateningly, I may allow them to observe me from a short distance, even interact briefly, my suckers tasting their wetsuit or glove, evaluating the strange creature that does not fit into any category I recognize. Humans are fascinated by this interaction. They interpret my color changes and my willingness to touch them as signs of communication or affection. The reality is more ambiguous. I am investigating, gathering information, responding to novelty. Whether I form anything like a social bond or recognition of individual humans is unclear. Some research suggests that octopuses in aquariums can learn to recognize individual caretakers and respond differently to different people, approaching those who feed them and avoiding those who have handled them roughly. This implies a capacity for individual recognition and possibly something like preference or aversion. But whether this occurs in the wild with divers who appear briefly and then vanish is uncertain. The ocean seasons are subtle compared to terrestrial seasons, marked more by temperature shifts and changes in current patterns than by dramatic transformations in the environment. But they exist, and I feel them. The water gradually warms as spring progresses towards summer. Food becomes more abundant as prey species reproduce and young animals become available. My growth accelerates. My behavior begins to shift as hormones associated with reproduction increase in my bloodstream, produced by the optic glands and other endocrine tissues. I become more active in seeking out females, following chemical trails more persistently, spending less time in dens and more time moving across the reef. I encounter males more frequently as well, and these encounters are now more agonistic. We display aggressively, and if neither backs down, we grapple, arms intertwining, suckers gripping, each trying to establish dominance without inflicting serious injury. I win some of these contests and lose others. Size is not the only factor. Motivation, health, and perhaps experience also play roles. When I lose, I retreat and wait for another opportunity. When I win, I continue searching for females. I find a receptive female in her den, and this time the interaction proceeds differently. She accepts my approach, her color patterns signaling readiness. I extend my hectocotylus, reaching carefully into her mantle cavity. The modified arm tip delivers spermatophores, packets of sperm enclosed in protective sheaths into her oviducal opening. The process is delicate. If I am too rough, she may reject me or attack. The mating lasts nearly an hour, during which time we remain in close contact, arms intertwined, colors synchronized. There is something almost hypnotic about the experience, a coordination of movement and physiology that goes beyond simple mechanical sperm transfer. When the mating is complete, we separate. There is no pair bond, no continued association. She retreats into her den, and I move away, my immediate reproductive drive satisfied but not extinguished. I may mate again with other females over the coming weeks. She will store the sperm I provided, and weeks or months from now, she will use it to fertilize her eggs when conditions are optimal. My own future is now limited. The hormones associated with reproduction are beginning to take their toll, triggering changes in my physiology that will eventually lead to senescence and death. My appetite begins to decline slightly, though I still hunt. My movements become less cautious. I take risks I would have avoided earlier, exposing myself to predators with less concern. This is not a conscious choice, but a shift in the neurological and hormonal systems that govern behavior. My body is prioritizing reproduction over survival, a strategy that makes evolutionary sense because I have already passed on my genes. I continue my routine, but the pattern is changing. I am less careful about maintaining my dens, less diligent about grooming, less responsive to minor injuries. A small wound on one arm, perhaps from a rocky encounter or a crab's pincer, does not heal as quickly as it would have months ago. My immune system, never particularly robust in cephalopods, is declining further. I am still functional, still capable of hunting and defending myself, but the trajectory is clear. I encounter a particularly large male, significantly bigger than I am, occupying a prime section of reef. Normally, I would avoid direct confrontation with such an individual, but my altered behavioral state makes me less risk-averse. I challenge him, displaying aggressively. He responds in kind, and we engage in a physical contest. His size advantage is overwhelming. His arms are longer, his suckers more numerous and powerful. He pins me against a rock, his suckers gripping my mantle and arms, restricting my movement. I struggle but cannot break free. The contest could turn lethal. He is capable of biting me with his beak, injecting venom, or simply holding me until I exhaust myself and drown. But octopus combat rarely escalates to lethal violence. After establishing dominance conclusively, he releases me. I jet away, my mantle contracting forcefully, expelling water and propelling me backward into a crevice. My pride, if I can be said to have such a thing, is damaged, but I am otherwise unharmed. I remain in the crevice for an extended period, my color pattern subdued, my activity level minimal. This might be interpreted as something like discouragement or defeat, though attributing such emotional states to an octopus is speculative. Eventually, I emerge and resume hunting, avoiding the territory of the dominant male. The hierarchy among male octopuses is loose and situational, not a rigid dominant structure like that seen in some social mammals. Dominant individuals control access to the best dens and the most females, but their dominance is constantly tested, and a male who is dominant in one area may be subordinate in another. The system is fluid, based on repeated individual interactions rather than stable long-term relationships. As weeks pass into months, the season shifts again. The water temperature peaks and then begins to decline. Prey abundance remains high through the summer but starts to decrease as autumn approaches. I continue to mate opportunistically, though my condition is declining. My weight has actually decreased slightly despite regular feeding, a sign that my metabolism is becoming inefficient, that my body is catabolizing its own tissues. My arms are slightly thinner, my mantle less firm. My skin shows signs of wear, small lesions and patches where the chromatophores no longer function properly. I am aging rapidly, compressed into months, a process that takes years or decades in longer-lived animals. I witness the reproductive process from the other side when I encounter a female guarding eggs. She is in a small cave, her body positioned at the entrance, her arms arranged around clusters of eggs that hang from the ceiling of the cave like bunches of translucent grapes. Each egg contains a developing embryo, visible as a tiny dark spot that grows larger each day. The female herself is visibly deteriorating, her skin pale and blotchy, her movements weak. She has not eaten in weeks, possibly months, and she will not eat again. Her eyes, once bright and alert, are clouded. When I approach, she rouses herself enough to display a weak threat pattern, but she lacks the energy to attack or flee. I do not threaten her eggs. I have no interest in consuming them, though some predators, including other octopuses, will raid nests if given the opportunity. I move past, leaving her to her vigil. Days later, I pass the same den and see that the eggs have hatched. Thousands of tiny paralarvae are drifting out of the cave, carried by the current, transparent and delicate, each one a miniature octopus measuring only a few millimeters. They pulse their mantles, expelling water, swimming upward toward the surface and the plankton-rich waters where they will spend the first phase of their lives. Most will die within days, consumed by planktivores or unable to find sufficient food. A few will survive to settle on the reef and begin the benthic phase, growing into juveniles, then adults. The female is dead, her body lying in the entrance of the den, already being consumed by scavengers. Her life ended with the hatching of her offspring, her biological purpose fulfilled. I am following the same trajectory, though I am earlier in the process. My mating season has been moderately successful. I have transferred sperm to multiple females, increasing the probability that some of my genes will appear in the next generation. But success is measured in probabilities, not certainties. I do not know how many of those matings will result in fertilized eggs, how many eggs will develop successfully, how many paralarvae will survive. The ocean is a harsh environment, and reproduction in octopuses is a strategy of overwhelming odds with sheer numbers rather than ensuring the survival of a few carefully tended offspring. Autumn deepens and the reef changes. Some fish species migrate to deeper water or to different regions entirely. Others become less active, their metabolism slowing in response to cooler temperatures. I too feel the cold more acutely now, my own metabolism sluggish, my movements slower. I hunt less frequently, my appetite diminished. I spend more time in my den, emerging only when hunger or restlessness drives me out. One evening I make a mistake, a lapse in the vigilance that has kept me alive this long. I emerge from my den without thoroughly checking for predators. A large grouper, perhaps the same individual I evaded months ago, is waiting nearby. It strikes the moment I am clear of the entrance, its mouth gaping, its body accelerating with surprising speed for such a large fish. I see it at the last moment and jet sideways, but I am not as fast as I once was. The grouper's jaws close on two of my arms, biting through the tissue, and I autotomize both limbs, severing them to escape. I jet away, ink trailing behind me, and wedge myself into a crack too narrow for the grouper to follow. I have lost two arms, one from the left side and one from the right, not adjacent but nearly opposite, which creates a balance problem. My remaining 6 arms can still function, still hunt and manipulate and defend, but my coordination is impaired, my grip strength reduced. The wounds where the arms detached are bleeding hemolymph, my pale blue blood, though the bleeding will stop soon as clotting factors activate. Infection is a significant risk. The ocean is full of bacteria, and my immune system is already compromised by age. The lost arms will regenerate over time, but I do not have time. My lifespan is measured in weeks now, not months. I remain hidden for the rest of the night and through the following day, my body working to seal the wounds and begin the early stages of regeneration. Small buds of tissue appear at the amputation sites, the first signs of new arm growth, but the process is slow, and I am weak. I eventually emerge to hunt because I must. Starvation will kill me as surely as predation. I catch a small crab enough to sustain me for a day, but the hunt is difficult with only 6 arms and I am clumsy, less efficient than before. The following weeks are a struggle. I adapt to my reduced number of arms, learning to compensate, but I am clearly declining. My skin lesions have worsened, patches of chromatophores no longer responding to neural signals, leaving permanent blotches of color that break my camouflage. My eyes are less clear, my vision slightly blurred. I am more vulnerable to predators, less successful at hunting, and my body is consuming itself to maintain basic functions. I encounter fewer octopuses now, whether because their numbers are actually lower or because I am moving less and therefore encountering fewer individuals is unclear. The reef seems quieter, though this may be a perception bias, my own declining awareness interpreting the world as less vibrant. I spend more time motionless, conserving energy, my arms loosely draped over rocks, my mantle barely moving as I breathe. A hurricane passes far offshore, its effects reaching the reef as increased surge and turbidity. The water is murky for days, filled with suspended sediment, and visibility drops to less than a meter. I remain in my den, riding out the disturbance. When the water clears, the reef has changed subtly. Some coral has been damaged, rocks have shifted, and the distribution of prey animals is different. I explore cautiously, mapping the changes, updating my mental representation of the environment. I find a dead fish, recently killed, perhaps by the storm surge smashing it against rocks. Scavenging is not my preferred feeding mode, but I am not in a position to be selective. I consume the fish, my beak tearing into flesh that is already beginning to decay. The meal provides energy, but it may also introduce bacteria or parasites that my weakened immune system cannot fight effectively. Time becomes less distinct. I am not sure how many days have passed since I lost my arms, since I last mated, since I hatched from an egg in the plankton and drifted down to the reef to begin my benthic life. My memory, never organized in a linear narrative fashion, is becoming fragmented, less reliable. I still know how to hunt, how to hide, how to navigate, but the details of specific events blur together. I encounter a female guarding eggs, and for a moment I feel a resurgence of reproductive drive, an impulse to mate again. But the impulse is weak, easily overridden by other priorities like finding food or avoiding a passing predator. My hormonal systems are in disarray, conflicting signals producing erratic behavior. I approach the female, but she displays aggressively, and I withdraw without challenging her. I am moving less now, spending days at a time in my den, emerging only briefly to hunt. My last successful hunt was a small shrimp, barely a mouthful, insufficient to meet my energy needs, but all I could catch. My arms are weaker, my reflexes slower, my coordination impaired. The regenerating arms are growing, now small stubby protrusions a few centimeters long, but they are not functional yet, just additional drains on my dwindling resources. One morning I do not leave my den. I remain inside, my body curled loosely, my arms tucked beneath my mantle, my eyes half closed. My breathing is shallow, irregular. My skin has faded to a pale, mottled pattern, no longer actively camouflaging but simply defaulting to a low-energy state. My 3 hearts are still beating, but the rhythm is erratic, occasionally pausing for seconds before resuming. I am dying, the inevitable end of the semilporous life cycle that characterizes all octopuses. My body, driven by hormones from the optic glands, has been systematically shutting down non-essential functions, redirecting resources to reproduction, even though the reproductive effort is now complete. There is no recovery from this process, no possibility of reversal. I am not experiencing pain in the way a human might understand pain, though I am certainly experiencing malfunction, the failure of systems that have operated reliably for my entire life. Whether this constitutes suffering is a question without a clear answer. A small, cleaner shrimp enters my den, its antennae twitching, investigating my body. Normally I would react, either eating the shrimp or expelling it. But I do not have the energy. The shrimp begins to pick at my skin, consuming dead tissue and parasites, performing its cleaning function even though I am no longer a viable host. Other scavengers will follow once I am fully dead, crabs and worms and bacteria, breaking down my soft tissues, recycling the nutrients back into the reef ecosystem. My contribution to the future is not my body, but the genes I passed on through mating, the spermatophores I delivered to multiple females, some of which will have resulted in fertilized eggs, some of which will have hatched into paralarvae, a tiny fraction of which will have survived to adulthood. Those individuals carry half my genetic material, combined with half from the females, creating new combinations, new variations for natural selection to act upon. This is the only form of immortality available to organisms like me. Genetic continuation through offspring I will never meet. The light in my den fades as my eyes close fully for the first time in my life. My mantle ceases its rhythmic pulsing. My heart stops. My neurons fire erratically for a few more minutes, residual electrical activity that fades gradually to nothing. The organism that was me, the individual octopus with a unique genotype and a unique life history, ceases to exist. My body remains, already beginning the process of decomposition, but the complex pattern of organization that constituted a living octopus is gone, dispersed into entropy. Within hours, scavengers have found my body. Crabs tear at my flesh with their claws. Bristle worms burrow into my mantle. Bacteria multiply exponentially, breaking down proteins and lipids. Within days, little remains beyond fragments of skin and the hard chitinous beak, which will persist longer, perhaps washing up on a beach eventually or remaining on the reef as a small fossil-like remnant, one of millions scattered across the sea floor, the only durable traces of organisms that are otherwise entirely soft-bodied. But even as my individual life ends, the species continues. The females I mated with are guarding eggs or have already released paralarvae into the plankton. Those paralarvae are drifting, feeding, growing, a tiny percentage of them surviving to settle on reefs and begin the benthic phase. Among those survivors may be individuals carrying my genetic material, my particular alleles for color-changing speed or problem-solving ability or disease resistance, or any of thousands of other traits. Those traits will be tested by the environment, by predators and prey and competitors and pathogens, and some will prove advantageous, increasing in frequency in future generations, while others will prove neutral or deleterious and disappear. This is evolution, the ongoing process that has shaped octopuses over millions of years, transforming an ancestral mollusk with a shell into a soft-bodied, intelligent, shape-shifting predator. The octopus lineage stretches back at least 300 million years to the Carboniferous period, when the earliest cephalopods were diversifying in ancient seas. The loss of the external shell, which occurred independently in octopuses and in some other cephalopod lineages, was a pivotal event, trading the protection of armor for increased mobility and flexibility. This allowed octopuses to exploit new ecological niches, squeezing into crevices to hide from predators and pursue prey, adopting a lifestyle that favored intelligence and adaptability over defensive structures. The evolution of octopus intelligence is still poorly understood. Large brains are metabolically expensive, requiring substantial energy investment, and they only evolve when the benefits outweigh the costs. In mammals and birds, intelligence is often linked to social complexity, long lifespans, and extended parental care that allows for social learning. Octopuses have none of these traits, yet we have evolved cognitive abilities that rival those of many vertebrates. The explanation likely involves the demands of our predatory lifestyle. We must handle diverse prey types with complex defenses, navigate structurally complex environments, and evade numerous intelligent predators, all without the benefit of armor or speed. Intelligence, in the form of behavioral flexibility and learning, provides a solution to these challenges that is more adaptable than hardwired instincts. The distributed nature of octopus intelligence, with substantial neural processing occurring in the arms rather than being centralized in the brain, may reflect the challenges of controlling a body plan with 8 semi-independent appendages, each capable of complex manipulation. Centralized control would require massive information transfer between the brain and the periphery, creating bottlenecks and delays. Distributed control, with each arm possessing its own neural circuitry capable of executing complex motor programs autonomously, solves this problem but creates a body with a fundamentally different organization than that of vertebrates. Whether this produces a different kind of subjective experience, a consciousness that is distributed rather than unified, is one of the most intriguing questions in comparative neuroscience and philosophy of mind. Octopus camouflage represents another evolutionary marvel, a system that integrates vision, neural processing, and muscle control to produce transformations that occur in less than a second. The chromatophore system, with each chromatophore individually controlled by motor neurons, allows for pixel-level control of color across the entire body surface. The addition of structural color cells, iridophores and leucophores, adds another layer of control, producing colors outside the range of the chromatophore pigments. The muscular papillae system adds three-dimensional texture, completing the illusion. This system evolved under intense predation pressure. Octopuses are soft-bodied and nutritious, attractive prey for numerous predators, and effective camouflage is often the difference between life and death. The evidence suggests that octopuses accomplish this camouflage despite having limited color vision, perhaps even being functionally colorblind by human standards. How an animal with single-channel vision can match colors accurately remains a subject of active research. Proposed mechanisms include chromatic aberration detection, where different wavelengths focus at different distances on the retina, allowing the visual system to extract color information from focus cues. Skin-based light sensing, where photoreceptors in the skin itself detect ambient light and drive color changes independently of the eyes. And template matching, where the octopus compares the overall brightness and spatial pattern of the environment to stored templates and applies the appropriate color pattern without directly sensing color. Evidence exists for all these mechanisms, and it is likely that octopuses use multiple strategies in combination. The evolution of octopus reproduction, with its characteristic semiparity, is linked to the physiological constraints of the cephalopod body plan. Rapid growth and high metabolic rates require substantial food intake, and the energetic demands of reproduction are immense. Females invest enormous resources in producing thousands of eggs and then guarding them for weeks or months without feeding. Males compete intensely for mating opportunities and invest energy in sperm production and mating behavior. The programmed senescence controlled by the optic glands ensures that adults die after reproduction, preventing them from competing with their own offspring for resources and avoiding the energetic costs of surviving through periods when conditions are unfavorable for reproduction. This life history strategy, living fast and dying young, is effective in unpredictable environments where adult survival from one breeding season to the next is uncertain. The diversity of octopus species reflects adaptation to a wide range of marine environments. My species, the common octopus, is a generalist, inhabiting coastal reefs, rocky shores, seagrass beds, and even harbors and marinas. in depths from the intertidal zone to 200 meters. Other species are more specialized. The giant Pacific octopus, the largest octopus species, reaches arm spans exceeding 6 meters and weighs up to 70 kilograms, inhabiting cold, temperate waters of the North Pacific. The mimic octopus of Indo-Pacific waters can impersonate other animals, contorting its body and arms to resemble flatfish, lionfish, sea snakes, and other species, using mimicry as both camouflage and defense. The blue-ringed octopuses, small species found in tide pools and coral reefs, are among the most venomous animals in the ocean, their saliva containing tetrodotoxin powerful enough to kill a human. Deep-sea octopuses, such as the Dumbo octopuses of the genus Octopuses, have evolved ear-like fins and live at depths exceeding 4,000 meters, in eternal darkness and cold, feeding on worms and crustaceans on the abyssal sea floor. Each species has its own ecological role, its own suite of adaptations, its own life history. But all share the fundamental octopus body plan: 8 arms, a soft body, a sophisticated nervous system, and a brief lifespan culminating in a single reproductive event. This body plan has been successful for hundreds of millions of years, surviving mass extinctions that eliminated many other lineages, adapting to changing ocean conditions, radiating into diverse habitats. The success is measurable. Octopuses are found in every ocean, from the poles to the tropics, from the intertidal to the abyss, occupying ecological niches as diverse as any group of vertebrates. Human understanding of octopuses has evolved considerably over time. Ancient observations were limited to what could be seen of octopuses in shallow water or caught by fishermen, leading to a mixture of accurate observations and imaginative embellishments. Aristotle, writing in the 4th century BCE, provided surprisingly accurate descriptions of octopus anatomy and behavior, noting the hectocotylus and its reproductive function, the ability to change color, and the intelligence of octopuses in escaping from containers. But he also reported inaccuracies, such as the belief that octopuses copulate through the hectocotylus inserting into the female's nasal passage, an error based on misunderstanding the anatomy. Medieval and Renaissance naturalists added little to the knowledge base, often repeating and embellishing ancient accounts. The age of scientific exploration, beginning in the 17th and 18th centuries, brought renewed study based on dissection and systematic observation. Naturalists described the anatomy in detail, identified species, and began to understand the basic biology. But the behavior and ecology of octopuses remained poorly known because most observations were of dead or dying specimens, and the living animals in their natural habitats were inaccessible. The development of diving technology in the 20th century revolutionized octopus research. Scientists could now observe octopuses underwater in their natural behaviors for extended periods. This revealed the complexity of octopus behavior, the sophistication of camouflage, the problem-solving abilities, and the ecological roles. Laboratory studies, using octopuses kept in aquariums, allowed for controlled experiments on learning, memory, vision, and neurobiology. The sequencing of the octopus genome in the early 21st century provided insights into the genetic basis of octopus traits, revealing extensive RNA editing, a process where the RNA sequence is modified after transcription from DNA, allowing octopuses to fine-tune protein function in ways most other animals cannot. This RNA editing is particularly prevalent in neural tissues, suggesting it plays a role in the evolution and function of the octopus nervous system. Current research on octopuses spans multiple disciplines. Neuroscientists study the octopus brain and distributed nervous system, as a model for understanding alternative forms of intelligence and neural organization. Roboticists draw inspiration from octopus arms to design soft robots with flexible, adaptable appendages. Materials scientists study octopus skin to develop synthetic materials that can change color and texture. Conservationists assess octopus populations and the sustainability of fisheries. Philosophers and cognitive scientists debate the nature of octopus consciousness and what it might reveal about the relationship between brain structure and subjective experience. The ethical status of octopuses is increasingly recognized. Many countries now include octopuses in animal welfare legislation, requiring humane treatment in research and aquaculture. This recognition reflects growing evidence that octopuses are capable of experiencing pain and distress based on their behavioral responses to injury, their possession of nociceptors, specialized neurons that detect tissue damage, and their ability to learn to avoid harmful stimuli. Whether octopuses experience pain in a subjective sense comparable to human pain remains uncertain, but the precautionary principle suggests they should be treated as if they do. Octopus aquaculture is a growing industry, driven by high demand for octopus meat and declining wild catches in some regions. But octopus farming raises significant ethical and ecological concerns. Octopuses are carnivorous and require high-protein diets, meaning farmed octopuses must be fed fish or other seafood, creating a net loss of food resources rather than a gain. The solitary nature of most octopuses and their tendency toward aggression and cannibalism make high-density farming difficult, requiring either individual tanks, which are expensive, or accepting high mortality rates. The short lifespan and semiparous reproduction mean farmed octopuses die shortly after reaching market size, limiting the efficiency of farming operations. And the ethical concerns about farming an intelligent, sentient animal in confined conditions are substantial. Some researchers and advocacy groups argue that octopus farming should not be pursued, that the ecological and ethical costs outweigh the benefits. The ecological role of octopuses extends beyond their direct interactions with prey and predators. As predators, we exert top-down control on prey populations, influencing community structure. Our foraging creates habitat complexity. The middens we build become microhabitats for small invertebrates and fish. Our bodies, when we die, provide pulses of nutrients to scavenger communities and to the microbial loop. Our paralarvae, drifting in the plankton, serve as food for planktivorous fish and connect benthic and pelagic food webs. Our presence or absence can have cascading effects through the ecosystem, influencing species composition and abundance at multiple trophic levels. Octopuses also interact with numerous other species in non-predatory relationships. We host parasites including copepods, isopods, protozoans, and helminths, which use us as habitat and food source. Some of these parasites are specialists, occurring only in octopuses or only in cephalopods. We are occasionally cleaned by cleaner shrimp and cleaner fish, which remove parasites and dead tissue, a mutualistic interaction that benefits both parties. We sometimes share dens with other species, including fish and crustaceans, in relationships that may be commensal, where one party benefits without affecting the other, or possibly mutualistic, though the details are poorly studied. The fossil record of octopuses is sparse because our soft bodies rarely fossilize. Hard parts, the beak and occasionally the stylet, a small internal remnant of the ancestral shell present in some species, are occasionally preserved, and these fossils provide glimpses into octopus evolutionary history. The oldest definitive octopus fossil dates to the Carboniferous, approximately 325 million years ago, though molecular clock estimates suggest the octopod lineage may have originated even earlier. Fossil impressions preserving soft tissue are exceptionally rare but extraordinarily valuable. A Jurassic fossil from Germany preserves an octopus with arms, suckers, and ink sac visible, providing direct evidence of body-plan stability over nearly 200 million years. The relationship between octopuses and humans is likely to evolve further as both populations grow and overlap increasingly. Coastal development brings human activity into octopus habitats, with both positive and negative consequences. Artificial structures, piers, breakwaters, and sunken ships can provide additional den sites, but they also bring pollution, noise, and disturbance. Fishing pressure continues to increase in many regions, driven by growing human populations and demand for seafood. Climate change is altering ocean conditions in ways that will affect octopuses directly and indirectly, through temperature changes, acidification, deoxygenation, and shifts in prey availability. Some octopus populations may benefit from these changes, at least in the short term. Warming waters may expand the range of warm-adapted species into previously cooler regions. The decline of some predatory fish due to overfishing may reduce predation pressure on octopuses. Our short generation times and high reproductive output allow rapid adaptation to changing conditions. But there are limits to resilience. Extreme warming can exceed thermal tolerance, particularly for species already living near their upper thermal limits. Acidification may impair egg development and larval survival. Loss of complex reef habitats reduces den availability and prey diversity, and the cumulative effects of multiple stressors may produce nonlinear declines that populations cannot recover from. Conservation efforts for octopuses are complicated by the lack of basic data for many species. Population sizes, trends, and distributions are poorly known for most octopus species outside of commercially important populations. Life history parameters, habitat requirements, and ecological roles are understood in detail for only a handful of species. Without this information, assessing conservation status and designing effective management strategies is difficult. Efforts are underway to fill these knowledge gaps, using a combination of traditional survey methods, electronic tagging, genetic analysis, and citizen science observations from divers and fishermen. One promising approach is marine protected areas, regions where fishing and other extractive activities are restricted or prohibited, allowing ecosystems to function with reduced human impact. Studies have shown that octopus populations often increase inside marine protected areas, suggesting that fishing pressure is a significant limiting factor. Protected areas also preserve habitat complexity and maintain diverse prey communities, indirectly benefiting octopuses. However, the effectiveness of marine protected areas for octopus conservation depends on their size, enforcement, and connectivity, because octopus paralarvae disperse widely, and larvae produced inside a protected area may settle far outside its boundaries. The cultural significance of octopuses varies widely among human societies. In Mediterranean cultures, octopuses are a traditional food, featured in numerous regional dishes and fishing practices. In Japanese culture, octopuses, called tako, are consumed fresh, dried, and in sushi and takoyaki, and they appear in art and folklore as symbols of flexibility and intelligence. In Hawaiian culture, the octopus, hia, is associated with the god Kanaloa and appears in traditional stories as a shapeshifter and trickster. In Western popular culture, octopuses are often portrayed as mysterious or menacing, from Jules Verne's giant squid, often misidentified as an octopus, in 20,000 Leagues Under the Seas. to more recent depictions in films and literature emphasizing their alien intelligence. Scientific interest in octopuses extends to astrobiology, the study of life beyond Earth. Octopuses are sometimes invoked as examples of how intelligence and complex behavior can evolve in body plans radically different from those of humans and other vertebrates. If life exists elsewhere in the universe, it may have evolved along entirely different trajectories, producing organisms with cognitive abilities but different neural architectures, sensory systems, and body plans. Octopuses provide a terrestrial example of such alternative intelligence, demonstrating that sophisticated cognition does not require a centralized brain, a long lifespan, or social learning. This has implications for how we might recognize intelligence in extraterrestrial life, suggesting we should not assume that alien intelligence will resemble human intelligence in its organization or expression. The study of octopuses also contributes to fundamental questions in biology and philosophy. The evolution of intelligence in octopuses and vertebrates represents a case of convergent evolution, where similar solutions, in this case, complex nervous systems capable of learning and flexible behavior, evolved independently in response to similar ecological challenges. But the differences in how that intelligence is implemented, centralized versus distributed, social versus solitary, long-lived versus short-lived, reveal the multiple paths evolution can take to produce adaptive complexity. This informs our understanding of the relationship between structure and function, showing that there is not a single optimal design for intelligence, but rather a variety of designs, each with advantages and constraints. The question of consciousness in octopuses is perhaps the most philosophically challenging. Consciousness, defined minimally as subjective experience, the property of there being something it is like to be an organism, is difficult to study even in humans and impossible to access directly in other species. We infer consciousness in other animals from behavior, brain structure, and evolutionary relationships, but these inferences are always tentative. Octopuses behave in ways that suggest subjective experience. They avoid harmful stimuli in flexible ways that suggest more than simple reflexes. They explore novelty in ways that suggest curiosity. They solve problems in ways that suggest understanding. And they exhibit what appear to be sleep states possibly associated with dreaming. Their brains contain structures analogous to vertebrate learning and memory centers, though built from different cellular components and organized differently. But whether these features constitute consciousness is uncertain. It is possible that octopuses are what philosophers call zombies, organisms that behave as if they are conscious but lack subjective experience, all their behavior the result of sophisticated but non-conscious information processing. It is also possible that octopuses are conscious, but their consciousness is so different from human consciousness, organized so differently and associated with such different sensory and motor capacities, that it is fundamentally incomprehensible to us. The question may ultimately be unanswerable with current methods, but the attempt to answer it forces us to confront the limits of our understanding and the assumptions we make about the nature of mind. As I described earlier in this narrative, my own life has now ended. The individual organism that was me has ceased to function, its components returning to the environment, its patterns dissolving into entropy. But the genes I passed on persist, carried in the offspring that resulted from my matings, and through those offspring my genetic material may influence future generations, continuing to be tested by natural selection, continuing to evolve. This is the immortality available to organisms like me, not individual persistence but genetic continuation, the propagation of information across generations. The octopus species, Octopus vulgaris, will continue long after my individual death. New individuals hatch, grow, reproduce, and die in endless cycles, each generation slightly different from the last, adapting to changing conditions, filling the ecological roles that octopuses have filled for millions of years. The species is not a fixed entity but a dynamic population, a flow of genes and traits through time, shaped by mutation, recombination, selection, drift, and migration. As long as suitable habitat exists, as long as prey are available, as long as environmental conditions remain within tolerable ranges, the species will persist. But species too have lifespans, measured in millions of years rather than months. The fossil record shows that most species eventually go extinct, replaced by descendant species, or by unrelated lineages that evolve to fill similar roles. The average lifespan of an invertebrate species is difficult to estimate but likely on the order of 1 to 10 million years. Octopus vulgaris may persist for a significant fraction of that time, or it may split into multiple descendant species as populations in different regions diverge genetically, or it may go extinct, unable to adapt to environmental changes that exceed its physiological limits. The fate of the species depends on factors operating at timescales far longer than any individual life, factors that individual octopuses cannot influence or even perceive. At the largest scale, the cephalopod clade, the group that includes octopuses, squid, cuttlefish, and nautiluses, has persisted for over 500 million years, since the Cambrian explosion when most major animal body plans first appeared in the fossil record. Cephalopods have survived multiple mass extinctions, events that eliminated the majority of species on Earth, by adapting to new conditions, by diversifying into new ecological niches, by evolving new body plans and life strategies. The external shell, ancestral for mollusks, was reduced or lost multiple times independently in different cephalopod lineages, allowing the evolution of fast-swimming pelagic forms like squid and flexible benthic forms like octopuses. The evolution of sophisticated eyes, complex brains, and advanced behaviors allowed cephalopods to compete with fish and other active predators, occupying roles that might otherwise have been dominated by vertebrates. The future of cephalopods, including octopuses, depends on the future of the ocean. The ocean is changing rapidly due to human activities, warming, acidifying, losing oxygen, accumulating pollutants, and being depleted of fish and other resources. These changes are occurring at rates unprecedented in recent geological history, faster than many species can adapt. Some species will go extinct, unable to cope with the new conditions. Others will shift their ranges, moving toward the poles as tropical and temperate waters become too warm, or into deeper water to escape surface warming. Still others may adapt in place, evolving new tolerances, new behaviors, new physiologies, though such adaptation requires genetic variation and time, and the pace of change may exceed the capacity for adaptive evolution. Octopuses, with their short generation times and high fecundity, may be better positioned to adapt than longer-lived species like marine mammals or sea turtles. But adaptation has limits, constrained by the available genetic variation, by trade-offs where adapting to one stressor reduces tolerance to another, and by the speed and magnitude of environmental change. If changes are too rapid or too extreme, even adaptable species can be overwhelmed. The role of humans in determining the future of octopuses is substantial. Human decisions about carbon emissions, fishing regulations, coastal development, pollution control, and marine conservation will shape the ocean environment that future octopuses will inhabit. Humans have the capacity to mitigate the worst impacts, to protect critical habitats, to manage fisheries sustainably, to reduce pollution, and to slow climate change. Whether humans will make those choices, will prioritize long-term environmental health over short-term economic gains, remains uncertain. Octopuses, for our part, simply live, generation after generation, responding to the environment as we find it, adapting when adaptation is possible, going extinct when it is not. We have no agency over the large-scale forces shaping the ocean, no capacity for collective action or long-term planning. We are shaped by evolution, not shapers of evolution, subject to natural selection, not directors of it. Our intelligence, remarkable as it is, operates at the individual level, solving immediate problems, learning from individual experience, but not accumulating cultural knowledge or coordinating across generations. Yet in our individual lives, in the way we hunt and hide, solve problems and evade predators, change color and shape, we embody principles that transcend any individual. We demonstrate the power of flexible behavior, the advantage of learning over rigid instinct, the effectiveness of distributed control systems, the possibilities inherent in soft-bodied designs. We show that intelligence can evolve along multiple paths, that consciousness, if we possess it, need not be organized like human consciousness, that sophisticated cognition can exist in organisms with lifespans measured in months rather than decades. These principles, these demonstrations, these living examples are octopuses, contribution to the larger story of life on Earth. We are one experiment among millions that evolution has conducted, one solution among countless solutions to the problem of survival in a complex, competitive, ever-changing environment. Our experiment has been successful for hundreds of millions of years, and it may continue for millions more, or it may not. The answer depends on factors far beyond any octopus's control or comprehension. I, as an individual octopus, am gone now, my brief life complete, my small role in the vast ongoing process of evolution fulfilled. I hunted and hid, learned and adapted, mated and passed on genes, lived and died, as octopuses have done for countless generations and will do for countless more. My life was unremarkable in most ways, typical of my species, neither unusually successful nor unusually unsuccessful. I survived to reproductive maturity, mated successfully, and died as all octopuses die, of senescence following reproduction. I was, in essence, an average octopus, and in that averageness, I represent my species, the common octopus, and through my species, the remarkable and ancient lineage of octopuses that have inhabited Earth's oceans for hundreds of millions of years. The ocean continues its cycles, indifferent to individual lives, but built from individual lives, each one a thread in the larger tapestry. The reef where I lived will host other octopuses, some perhaps carrying my genes, all carrying the evolutionary legacy that produced the octopus body plan and lifestyle. The sun rises and sets, tides ebb and flow, seasons turn, and life, in its myriad forms, continues. This is the story of one octopus, one small part of one species, one brief moment in the long history of life in the sea. It is a story that has been repeated billions of times in the past, and will be repeated billions more in the future. Each iteration unique in its details but similar in its broad outlines. The endless repetition with variation that is the essence of life on Earth.